Like most people who get into that sort of cell and gene therapy space, they probably have some sort of personal story. For me, I had an uncle who was super important to me, and he came down with Parkinson's disease when I in my early 20s. It was really that guy, when I saw him just debilitate over the years, who drove my desire to one day get into making impact on people's lives with cell and gene therapy and making impacts for finding a cure, for finding not only Parkinson's disease, but any other debilitating disease like that, because just seeing it firsthand and how hard it can be on families, it made a huge impact on me. My name is Travis Hardcastle. I am a senior product manager for the engineered cells portfolio for Synthego Corporation. Gene editing can be hard, it can be difficult. We know that anytime we're affecting the genome from a CRISPR-Cas9 gene editing perspective, that there are possibilities that you're going to create some sort of off-target effect, right? Making sure your structural integrity with the genome is intact is extremely important, particularly as you're looking to make discoveries and pass that off to the clinic. Anytime you can make a contribution from a biotech standpoint into the clinic, I think that is success. I think that's ultimately success. You look at the different methodologies out there, whether it's, G-banding, right, which is the old school sort of, methodology for doing that. It's very time consuming. It takes specialized skill labor. You can look at ddPCR, right, which is great and you have a great turnaround time, but it's very targeted, right? There hasn't been, at least at this point, a catch-all that solves all those needs. It has high throughput, high speed to delivery, short turnaround times, as well as that captures all of the genomic abnormalities that you're looking for whenever you're doing these editings. You're trying to have that solution that suits everybody's needs and is able to capture all of the abnormalities and all the the variations that come with the genomic integrity. The level of investigation you want to have for looking at that genomic integrity is vastly more important as you move closer, I think, to the clinic. That's where optical genome mapping comes into play, right? Any of that in-depth QC that we need to do, we use a more detailed platform such as optical genome mapping. Using the optical genome mapping, we've been able to uncover some abnormalities that we weren't aware of before we started using it. And so it's allowed us to go back and now, from a manufacturing standpoint, subclone some of these populations to make sure we do have really high quality cell lines that we can provide customers with. It's a method and it's a technology that gives you a whole map of the genomic sort of abnormalities or the integrity of the genome that is not targeted like certain methodologies and is not time consuming like other methodologies. And I think it gives you the throughput, the capabilities, the things that you're looking for with a methodology. I think it offers a ton of advantages that the other methods can't provide. Array-based technology takes weeks to get turnaround times on. ddPCR technology, sure, 3-4 day turnaround time, but you're only getting targeted hotspots. So how can you get the best of all those worlds and combine them down into one? So I think that's where BioNano comes in. It's prime for change in the market. It's prime for a new technology to come around that's able to pull everything together that customers want and need in a scalable way, in a cost-effective way. And I think that's what BioNano is doing. I think that the future's bright.